A patient can walk into a clinic appearing physically fit and asymptomatic while harboring advanced, silent pathology. Clinical screenings routinely uncover a dangerous profile beneath these healthy exteriors: blood pressure in the hypertensive range, elevated fasting glucose, and LDL cholesterol levels high enough to trigger a vascular event. This damage follows a clear epidemiological timeline. Evidence from thousands of autopsies indicates that subclinical atherosclerosis begins in childhood for those raised on a standard American diet. Outward physical appearance is an unreliable diagnostic tool. Actual health status is determined by microscopic cellular biomarkers. This understanding has led to a clinical consensus shared by over 17,000 physicians and former leadership of the American College of Cardiology. They prescribe the whole food plant-based protocol as a targeted molecular-level medical intervention. While standard pharmacological treatments manage symptoms, a plant-based intervention addresses the underlying disease. Removing animal products allows the endothelium to recover and the body to begin repairing the damage. Peer-reviewed research confirms this protocol can arrest chronic disease, alter gene expression, and mitigate global ecological collapse. Treating the root cause of systemic illness requires a shift to evidence-based biochemistry. Coronary artery disease is driven by a specific mechanism of injury. Animal fats and proteins directly damage the endothelial cells, suppressing their production of nitric oxide, the molecule responsible for maintaining flexible, open arteries. This process involves specific dietary precursors. Choline, found in high concentrations in eggs, and carnitine, found in meat, enter the digestive tract. In the gut, bacteria break these compounds down into a byproduct called TMA. The TMA then travels to the liver, where it is oxidized into a compound called TMAO. TMAO embeds into the vascular wall, driving aggressive systemic inflammation. This chemical damage extends into oncology. High concentrations of blood choline and TMAO are linked directly to the progression of lethal prostate cancer. Harvard researchers followed more than 1,000 men with early-stage prostate cancer and discovered that consuming even less than 1 egg per day doubled the risk of the cancer spreading. Animal-based foods function as active biological precursors to toxic, oncogenic metabolites. Animal protein ingestion also upregulates the production of insulin-like growth factor 1, or IGF-1. High levels of IGF-1 act as a growth hormone for malignant tumors, driving their acquisition and progression. However, data from the Cleveland Clinic proves that these processes can be halted. Patients with severe heart disease who followed a strict plant-based protocol achieved angiographic reversal of their atherosclerotic plaque, a result surgery cannot replicate. This clinical success aligns with historical autopsy data. When comparing residents of St. Louis, Missouri, to those in rural Uganda, researchers found a stark contrast in cardiovascular disease. St. Louis autopsies revealed 136 myocardial infarctions, while the Ugandan group, matched for age and gender, had only 1. The Ugandan population was immune to coronary artery disease because their diet was centered almost exclusively on whole plant foods. Reversing arterial disease requires the absolute elimination of animal proteins to stop the continuous injury to the endothelium. Scaling these dietary choices up from the individual to the global reveals an identical pattern of systemic inefficiency. Animal agriculture occupies 83% of all agricultural land on Earth, yet it produces less than 20% of global consumed calories. The United Nations identifies this sector as a primary driver of land degradation, water pollution, and the loss of global biodiversity. Even grass-fed systems, often presented as a sustainable alternative, fail when scrutinized against environmental data. Grass-fed cattle generate higher methane emissions than feedlot cattle because of the chemistry of enteric fermentation. The massive land requirements for grass-fed beef make it an ecological impossibility to scale for the global population. Animal agriculture acts as a resource-intensive system that yields a disproportionately low caloric return for the land it consumes. This inefficiency is measured by carbon opportunity cost, the sequestration potential lost when land is used for livestock instead of native ecosystems. This system carries a dual penalty: the methane produced by the animals and the lost carbon sink of the destroyed forest. Reconfiguring current cropland to grow human edible plant foods exclusively provides a massive environmental rescue. Oxford data shows that in the US alone, this transition would feed an additional 350 million people. This would free an area of land equivalent to the US, China, Australia, and the EU combined for carbon-sequestering reforestation. Shifting to plant-based nutrition simultaneously neutralizes a leading source of emissions and maximizes global carbon capture. A plant-based diet also offers direct genetic benefits, specifically the lengthening of telomeres, the protective caps on your DNA. Clinical trials measured these genetic changes in patients after just 8 weeks of strict plant-based adherence. Both cellular health and ecological recovery depend on the same foundational choice: the removal of animal products from the system. Many clinical settings still recommend moderation, suggesting patients eat less meat or lean meats. Moderating a biologically incompatible fuel source does not prevent disease. It only reduces the speed of physical degradation. The scientific consensus across cardiology, oncology, and climatology points to a single solution. To address root causes, the data requires a complete shift to whole food plant-based nutrition. Human metabolic health and planetary ecological survival ultimately function as two outputs of the exact same scientific equation.